So our next speaker is Dr. Danny Miller. He's an assistant professor in the Department of Pediatrics, Division of Genetic Medicine, and the Department of Laboratory Medicine and Pathology at the University of Washington, and is an attending physician at Seattle Children's Hospital. His lab is developing long-read sequencing-based clinical genetic tests with the goal of increasing the rate of genetic diagnoses, reducing the time required to make a genetic diagnosis, and lowering barriers to obtaining comprehensive clinical testing. Clinically, he cares for patients in both general genetics and skeletal dysplasia clinics. So I think he'll explain in his talk what long-read sequencing is as well. So I'll leave it to him.
Thank you, Eric. I think you should do 2 truths and a lie when you do these introductions and let the group figure out which one's which. So thank you all for the opportunity to speak here. I really enjoy it. I've been to several family meetings now, and as a geneticist, I'll just say that every meeting I go to, I learn way more than I ever did in residency about any specific condition, just because you see so many folks in one spot. So thank you for the opportunity. I'm going to talk about a project that we have to basically build a resource for the rare disease community by collecting samples from individuals with particular conditions, including Kleefstra syndrome. So just to orient you, you're all made up of about 1 to 10 trillion cells, depending how you want to count. Every one of those cells has 2 copies of your DNA on chromosomes, and then, let's see, does the pointer work? It's kind of faint. Can you guys see that? Okay. The chromosomes are made up of DNA, of course, and then on that DNA are the genes, and the genes are the things that we probably know most well, and specifically in this room we know about EHMT1, the gene that's associated with Kleefstra syndrome. But that's not the whole story. Your brain cells need to know how to be brain cells, and your skin cells need to know how to be skin cells. And one way that they do that is by a process called DNA methylation. So your genes are just A's, G's, C's, and T's. They're just 4 letters. The DNA is modified by a process in your cells that puts little molecules or little marks on the DNA that says, I want you to use this here, or I don't want you to use this in a different spot. So that's DNA methylation. That's what tells the cell, hey, I want you to actually make this particular protein. So again, that's how your skin cells know how to be skin cells, or your, let's say, bladder cells know how to be bladder cells, or your heart cells know how to be heart cells. So why are we interested in DNA methylation? Why am I talking to you about that? Well, we know that certain genetic conditions have unique patterns of DNA methylation. So you have about 3 billion DNA molecules in your genome, and the methylation marks are at different spots in your genome. And certain diseases or certain conditions have different patterns of where those spots are, and Kleefstra syndrome is one of those. Knowing methylation patterns associated with unaffected and affected individuals can help us in a couple of ways. One way is it actually helps us diagnose individuals with a condition. It could be that you have genetic testing done and it doesn't find any spelling changes in your DNA that tells you, yeah, this is the particular condition. But we can look at the DNA methylation and it could say, oh, this is actually associated with this specific condition. The other thing is that it helps us better understand uncertain DNA changes. Did anyone in the room, when they got their diagnosis, have an uncertain variant in EHMT1 where they said, we see a spelling change, but we don't know for sure that it's associated with the condition? So it does happen. And what DNA methylation lets us do is it lets us ask a different way, could this be causing this particular condition? The final thing, and this is a bit speculative, is that methylation could change in response to treatment or therapies. And as I've gone to these meetings with groups that are developing new gene therapies or new ways of treating conditions, they're looking for objective ways to measure the outcome of those therapies. And I think DNA methylation could be one of those objective measures. But the problem is that today we don't have enough data, we don't have enough resources to actually detect and then measure those changes. So as Eric mentioned, my lab does something called long-read sequencing. The easiest way to think about this is it's basically every genetic test that you can do rolled into one. So instead of having someone look at your chromosomes using a karyotype, looking at whether or not there's a big piece that's added or deleted using a microarray, or just reading the genes themselves using sequencing, we do it all with a single test, and that's this long-read sequencing technology. So essentially the idea is that instead of it taking multiple years to get to a diagnosis, we can do it within a couple of days and even a couple hours. We sequenced a kid in my lab one night in 3 hours to figure out what it was that he had. So we technically have the world record for fastest diagnosis. I just don't have the time to apply for the world record. This is a movie that probably needs to hit play. I always think it's fun to look at the technology. Could you hit play on the picture, please? Oh, is it over here? So there's a couple of different ways that we can do this thing called long-read sequencing. I think it's just kind of fun to see what it looks like. We mostly use a technology called nanopore sequencing, and all it's doing is it's measuring changes in current as DNA molecules go through these tiny pores. So we can use that current change to work backwards to figure out what the sequence is of the particular molecule. The neat thing about this is that it's available almost in real time. So as we're sequencing, we actually know what DNA is there. And not relative to Kleefstra, but we're using this technology to develop ways to diagnose diagnose tumors in the operating room, like during surgeries. So this actually works in almost real time. Okay, so I'm just going to talk about quickly how we use DNA methylation with long-read sequencing, and I'm going to show you some data. I find, as I've done more of these, I find that families really appreciate seeing actual data. So that's the way I've been going with these talks. These are two plots. There's a heat map over here on the left and what we call a PCA plot on the right. And this is from a published paper, and I'm sorry, the citation got cut off. Basically what they're showing is that individuals in green are the controls. They're individuals who don't have a condition. Individuals in blue are those that do have a particular condition. In this case, it's something called Cornelia de Lange, and then again, the controls are just in green. So what you can tell is that the green is all together, and the blue is all together, so they've grouped. So they've used like a statistical method to basically say, these group of individuals are more similar than the other group of individuals. The two red dashes are individuals who have uncertain variants, who are thought to have the condition, but the variants, they're not sure if they actually caused the condition. So using DNA methylation, what we can do is we can say this particular individual groups with the controls, so we might say that they don't have the condition, whereas this other individual on the left groups with the individuals who are affected, so we would say they do have the condition. So at the most basic level, this is how we can use DNA methylation to help understand whether or not someone has a particular genetic condition. The fun thing about, really the fun thing about data is that you can look at it in multiple ways. So you can transform this data into these plots, these PCA plots, and you can look at them in different ways. So there's a DNA methylation associated with about 200 or so conditions, and unfortunately most of the data and methods associated with those are not publicly available. They're held by a private company. So I can't take sequence data from anyone in this room or anyone with any genetic condition and compare it with all the data that's been generated before. There's a small amount of data that's available. It's not available for Kleefstra syndrome. It's available for some other conditions. So the goal of our lab, the work we're doing, is to make these resources, to generate the data, and to make the resources publicly available, which is why I'm standing here. This is just an example showing you that we can do it. So this is a PCA plot from data that we got from some publicly available data for a condition called Kabuki syndrome. You heard Anna mention it previously. The green dots are kiddos who do not have Kabuki, and the blue dots are kiddos who do have Kabuki. And you can see that they separate, which is exactly what we want. So this is work done by Sophie Gibson, who's a PhD student in my lab. She's absolutely fantastic. She can take long-read sequencing data, and she can separate control individuals from individuals who have Kabuki syndrome, the triangles, using some of the published data. So we know that this works. We can build specific, Sophie's been able to build classifiers, so build methods that separate just long-read derived data really well. So for example, the controls are here on the right, the cases are on the left. This is for a different condition called SETD1B, but essentially it's saying that we know that it works. And we're writing a tool, we've written a tool called EpiSignaler, where you could go online, download this, and use this to analyze data. So any researcher throughout the world could use this to analyze their data. So my goal is that we're going to build something similar for Kleefstra syndrome, using samples collected, hopefully here. We get some samples from Combined Brain, and then we can also collect samples over by mailing you kits. So what do I mean by that? So I collect blood using a device called a Tasso. It's a small device that you stick on your arm. It pulls out about half of a mil of blood. Does anyone have a continuous glucose monitor or has seen those CGMs that they put on your arm? Exact same idea. Makes a tiny cut, pulls out about half a mil of blood. It takes about 2 to 8 minutes depending on the person. Some of us bleed really quickly, some of us are a little bit slower. I'll say I've done, I don't know, 10 of these at this point. Most kids, they're just scared of the idea, and once you start doing it, they ask you when you're going to start, because they have no idea that you actually started pulling the blood out. I also have cheek swabs that I'll try to collect. I don't know if I'll be in a particular room or just out in a hallway. I don't know if we decided yet, but I think TBD.
So you're going to be in Livermore, which
Okay.
is right beside where Simon's is. So
Okay.
if people are already going by Simon's, they can stop in to see you as well.
Great. I'll be here the rest of today and then all day tomorrow as well. I'm not leaving until Sunday. So please stop by and say hi. I will say that if you are interested in participating but can't do it while you're here, we can actually mail kits to your house, and you can mail them back to us. So you can do these at home as well. And my email is here, and again, I'll be in the room, so just come and talk to me too. And that's all I have. I'll say thank you, and I'm of course happy to take any questions, or just talk about sequencing in general, because I think it's really cool.
Any questions for Dr. Miller? Oh, okay.
Thank you very much. It's very scientific and a little bit beyond my understanding. But question, you said that a lot of information belongs to private companies and not publicly available. Because it's for greater good, what are the means to make it publicly available, or just new rules and regulations and functioning under these new rules and regulations?
Yeah, without getting into, I get a little worked up on this, so I apologize. The way that this group, when they write a paper, they say that the data can't be shared because of institutional, they work at a university, and they say it can't be shared because of institutional limitations, which is bogus because, I go to my IRB and I say I'd like to share the methylation data publicly, and, I have to answer the right questions, but they're like, of course you can do that. So it's not this can't be done, it's they don't want to do it because they make money off of it. So it really irritates me and other researchers that we don't have access to that data. So how do we get around it? Participate in studies like this where the data is going to be shared, released publicly.
Just statistically, it will have access. It will give access to so much more to build more research.
Yeah.
So I understand
exactly.
the sentiment.
Yeah. Yeah.
Thank you.
Yeah.
Any other questions? Jeff has a question.
Uh-oh.
No, I guess I just want your feedback. We see a lot of folks who will come into our community and talk about VUSs. And I'm just wondering if you have any kind of estimation of what that could do as far as population growth, because a lot of what we hear about is, there's an estimated prevalence of 10,000 kids, and there's, maybe 400 that we've identified in the U.S. What could something like this, or just better understanding of VUSs, mean for our community and the growth and identifying patients?
Yeah, well, with sequencing-based newborn screening also, I think you're going to find a lot of individuals identified that may not have been identified previously. I think what it will do is, I think it'll help clarify, maybe it'll help stratify and clarify some of the how severe some of the individuals are affected with methylation. We may be able to predict, this individual might have these features, whereas this other individual may not. Really what I guess my 5 years out hope is that there just are no more uncertain variants. In other words, you see someone who comes in and says, I have an uncertain variant in EHMT1 or even EHMT2, which we heard about in the scientific sessions earlier. It kind of really clarifies that and doesn't give, I've met a lot of families who have been involved in the community and then end up getting the diagnosis taken away, if you will, and it hopefully prevents some of that as well.
Okay.
Thank you, everybody.
Thank you. Our next speaker